guys, and welcome to AP Help 101. So today I'm going to be talking about photosynthesis, and that is just as important as cellular respiration, and it comes a lot on the test. So let's get started. Just a brief introduction about photosynthesis. Basically what happens is light energy converts to chemical bond energy. That's, in basic terms, what photosynthesis is. And there are two processes. There's a light-dependent and a light-independent process. So we're going to talk about some of the photosynthetic pigments because they play a huge role in photosynthesis. They absorb light energy and they provide energy to carry out photosynthesis. And there are two groups of pigments. We have chlorophylls and carotenoids. Carotenoids. Man, that is really hard to pronounce. Carotenoids, I believe. So we're going to start with our chlorophylls. We have chlorophyll A and chlorophyll B, and they are green. So these pigments are green, and they absorb red, blue, and violet. Carotenoids, they are yellow, orange, and red color, and they absorb blue, green, and violet colors. Then there's other pigments as well. There's a xanthophyll, and that's a carotenoid, but with a slight chemical variation. We don't really need to know so much about it, but these are just examples of other pigments. We have phycobilins, which are reddish in color and they absorb blue and green. And we have antenna pigments, which actually is the name for chlorophyll B, carotenoids, and phycobilins. Photosynthetic pigments. Talking more about these actually. So there's an action spectrum, and it's a graph that you're probably going to need to understand. Basically what it is there are wavelengths of light plotted against the rate of photosynthesis, and actually later I have a picture and I can help explain this more. So we just need to know just a couple more facts about pigments. Chlorophyll A is used directly in photosynthesis. It has a porphyrin ring and a hydrocarbon tail, and that ring is actually the head of chlorophyll A. And the double bonds in the porphyrin ring are a source of electrons, and that's where we can get how we can make the ATP and the sugar in photosynthesis. So this is a graph. This is an action spectrum. At the top, you'll see an action spectrum, and on the x-axis, you'll see the wavelengths of light. So you see the violet, the blue, green, yellow, the colors of the rainbow. And then on the y-axis, you'll see the rate of photosynthesis, or absorption. Almost the same thing. So you can see that the more absorption there is, that means less is reflected. So if there's less of, for example, I believe chlorophyll, is that B? I think it's chlorophyll B. It comes at a low point around 550 nanometers. Which is the wavelength between green and yellow. So you can see that most of that color is reflected and not absorbed. So I think this is an important graph just to be able to read. It might come up on the test. Okay, we're just going to talk about the chloroplasts a little bit because that is where photosynthesis does take place. It does contain photosynthetic pigments, so the ones that we talked about, it does contain those. Light-dependent reactions take place in the grana. Light-independent reactions take place in the stroma, and the grana is made up of many thylakoids, or small discs, where photosystems 1 and 2 are located. So here's a picture of the chloroplast, and you can see the tiny discs inside. Those are the thylakoids, and a stack of those discs is called a granum, and many granums is grana. And of course, you can see the stroma right there, where on the inside of near the thylakoids. So we're going to talk about the photosystems now. There are light harvesting complexes in the thylakoid, and each has a reaction center which has chlorophyll A and a place where there are hundreds of antenna pigment molecules which give energy to chlorophyll A. So basically, there's a lot of moving parts in photosynthesis, and photosystems are one of the many things, so it has a reaction center, and then there's a place where these antenna pigment molecules, so those are your chlorophyll B, your carotenoids, and they're going to give that energy to chlorophyll A. And I'll explain more step by step what photosynthesis is. This is just a basic explanation of what all the parts are. And there are two types of photosystems. There are 1 and 2. That is, they were discovered in that position. So photosystem 1 actually is not the first photosystem. Photosystem 2 comes first, but it was discovered second. So photosystem 2, PS2, I'm going to short form photosystem as PS. And it works in a 680 nanometer range. And photosystem 1 works in the 700 nanometer range. So now we're going to talk about light-dependent reactions. So in light-dependent reactions, light is absorbed by photosystems and photosystems, by both photosystems, and electrons move down the electron transport chain. And there are two possible routes for electron movement: non-cyclic and cyclic photophosphorylation. Ooh, that's a big word. So we're going to talk about non-cyclic photophosphorylation. There are two electron transport chains, and in the end, we create ATP and NADPH, which is similar to NADH, which is in cellular respiration, but there's an extra phosphate here. So I have them labeled in steps. First, we're going to go to photosystem 2. Energy is taken in. The electrons in the head of the chlorophyll A excite, and they move to a higher energy state and are taken by a primary electron acceptor. So basically, what's going to happen is light hits the photosystem 2, and the electrons in the head of chlorophyll A are going to get excited, and they're going to shoot up. Basically, that's basically what happens. And they're taken, and then when they shoot up, it's taken in by a primary electron acceptor. Then photolysis takes place, splitting of water, and replaces electrons lost in chlorophyll A. So basically, this is just, we need to replace the electrons. So we're going to split some water, replace the electrons. So that way we can still send those electrons to the primary electron acceptor when light hits. Then we have the electron transport chain, where electrons from photosystem 2 go down an electron train, just like the one in cellular respiration, and that is where ATP is created by chemiosmosis. Again, same process as cellular respiration. Chemiosmosis, process that creates ATP. Protons are released from water, move to thylakoid space. ATP made when protons move from thylakoid space through ATP synthase channels into stroma. So again, I have the diagram of the chloroplast on the other slide, so you can always go back and look at the picture just to see the movement. But this is the process of what's going on basically. So it's almost the same as cellular respiration. There's a lot of similarities here. So that's chemiosmosis. We use it in both cellular respiration and photosynthesis. Again, there's more steps in non-cyclic photophosphorylation. NADP, it picks up two protons released from water, and then that NADPH, because now it's picked up those two protons, takes the hydrogen to the Calvin cycle to build sugar. And the Calvin cycle is the second part of photosynthesis. This is the first part. And finally, we're going to go to photosystem 1, which is basically the same process of photosynthesis, photosystem 2, which energy is taken in. Electrons in head of chlorophyll A excite, move to higher energy state, taken by primary electron acceptor. But then, the electrons are replaced by electrons from photosystem 2, and in the end, photosystem 1 produces NADPH. So photosystem 2 does not create NADPH, photosystem 1 does that. Photosystem 2 makes the ATP. Then we have cyclic photophosphorylation. And this produces ATP only, not NADPH, and no oxygen is released at all. So this is a shorter form of, instead of non-cyclic photophosphorylation, this is kind of a shortcut, really. So the electrons move from photosystem 2 to photosystem 1 to the primary electron acceptor and then back to photosystem 2. So basically, it doesn't go through the second half. After the electron transport chain, it doesn't go to that second half. It just goes straight from photosystem 2 to photosystem 1. There's no that electron chain. That's where your ATP is created, but then there's no second excitation of that electron. It just goes back to photosystem 2, and it just creates more ATP. And this is actually used more when the plant is just on a shortage of ATP and it needs to create some really fast. This is the fast mode of creating ATP. And the Calvin cycle. It's the second part of photosynthesis. It's also known as the light-independent reactions. Now, that does not mean that it only takes place during the night. No, photosynthesis takes place during the day. All of photosynthesis does. They just named it the light-independent reactions, and you'll see why. So in the end, it creates P-gal, which is a 3-carbon sugar. Carbon comes in, and it basically becomes incorporated into that P-gal. Basically, that's what happens in the Calvin cycle. Not much really happens. This process is called carbon fixation. So here's the longer steps. Carbon dioxide comes in and attaches to a 5-carbon sugar, making a 6-carbon sugar, and that becomes unstable, so it breaks down into 3 carbon molecules of 3-PGA. And again, it only occurs in the light. Only. It doesn't take place in the dark because it's called the light-independent reactions. But anyways, that is photosynthesis. That's basically what you need to know about it. If you have any questions or comments, leave them down in the comments below if you have any suggestions. And I'll see you guys next week.